About the handout that I just passed out to you, the beautiful color photograph that shows a picture of some cells. We're going to talk about one side of the picture first, and in doing so we'll actually have talked about most of the things that are on the other side. I think I might have put the holes in such a way that we think that we're going to look at the animal cell first, but we're actually not. We're going to look at this one, the one that has all the lines on it. Now last week I asked you to think about what you already knew about a cell. I asked you to think about a rectangle and a circle and which one would be which. This is definitely a plant cell. Plant cells have rigid edges or rigid looking sides as opposed to an animal cell, which is usually more circular in shape. A plant cell doesn't have to be a perfect square or rectangle, but it does have sort of straight line edges on it because of one of the pieces of its structure. So what we're going to do first is we're going to go through and we're going to name a bunch of the pieces that are on this diagram, and then we'll talk about their functions afterwards. We'll start right here at the top. This is the nucleus. And a lot of people last week when I looked at your sheet remembered that that was a part of the cell. So the big solid purple thing is the nucleus. Now keep in mind this picture is showing a cell that is cut open, and it's also showing a nucleus that is cut open so that you can see what the inside of it would look like. The nucleus isn't 3/4 of a circle, it's a whole round thing, but it's cut open in this picture so we can see the inside. Now, the nucleus itself has a few parts to it. One of them is this central dark portion of it. And so that second line of the 4 that are pointing to the nucleus is trying to point right to that central piece. This central dark portion is called the nucleolus. So it's like the nucleus of the nucleus. The other 2 lines are pointing to structures that are on the surface of the nucleus. This sort of film or envelope that is around the nucleus is sometimes referred to as the nuclear envelope or just simply the nuclear membrane. Either one is fine for me. And then on that nuclear membrane, the last line is trying to show one of the little holes that's on it, and that would be called a nuclear pore. So notice that all of the structures that are part of the nucleus start with the same few letters, N-U-C-L-E. So if you ever see the word nuclear, we are talking about the nucleus of a cell. Now around the nucleus, you have this wavy looking structure. And the way that you'll identify where this is in a cell is usually by finding the nucleus first. And so I'm pointing this out specifically. The structure that you're looking at is called the Endoplasmic Reticulum, which is often just abbreviated the ER. Now you'll notice that there are a couple of lines that point to this structure. The line that I'm indicating here is actually pointing to the rough endoplasmic reticulum. So we could call it the RER. And this line over here at the top is pointing to the smooth endoplasmic reticulum. And the way that you would know the difference between the rough and the smooth is whether or not these little dots are on it. So these little dots all over the place can be found on the endoplasmic reticulum, or they can be found just out in the cytoplasm of the cell. These little dots are called ribosomes. So for you to know whether you were looking at the rough or the smooth endoplasmic reticulum, you'll look for the little dots called ribosomes. The right side of the diagram has them, and the upper left side doesn't, and so that's how we know the distinction between them. Does anyone have any questions so far and need me to repeat any of the explanations on the diagram? Oh, we got orange. The next piece will come down here is this one. Now this, just like all of the other organelles, is cut so I can see inside of it. So most of these organelles just look like a sac when you look at them from the outside. But this has been sectioned so we can see the inside. And it is called a mitochondrion. If I wanted to say the plural version of this word, that's where I would say mitochondria. Now, I'm not the grammar police. I'm not going to say that you're wrong if you give me plural instead of singular, but just so you know, if there's one of them, it's a mitochondrion. If we're talking about a bunch of them, then we can say mitochondria. Now the mitochondria, when you look at it, is kind of an oval shape. And there's another organelle that's also kind of an oval shape that I'm going to point out right now. It's this one over here. This is your chloroplast. A lot of people remembered that plants had something that made them look green. Chloroplast is the name of the organelle. If I look at a chloroplast and a mitochondria, obviously in this picture a chloroplast is green. Will everything always be in color? No. And so when you look at the structure, see how a chloroplast has what looks like little stacks of pancakes on the inside, whereas a mitochondrion has a wavy line? That could be your visual cue for what the difference between them is. Now we'll come back over to the top here. This structure right here is called the Golgi Apparatus. You'll notice that it looks a lot like the endoplasmic reticulum, except that it's very visibly separate. It's not right around the nucleus. It's kind of off on its own a little bit. Now we have two examples of really the same thing. This line right here and this line right here are both pointing to a vacuole or a vesicle. It basically means a sac that's holding something. The difference between them, and why one is so big, is that the huge one is called the central vacuole, and it's for water. And we'll talk about how this central vacuole helps a plant out in a little bit. But plants will always have this really big vacuole in the center that's holding water. They can also have other smaller vacuoles all around, and that's because the vacuole's job is to hold things or store things. Now there are some other pieces on the inside. These three lines collectively here are referring to the cytoskeleton. I'm not going to ask you to name all of the different little subcomponents of it, but basically we're saying that there are little structural pieces inside of a cell, kind of like how you have a skeleton inside of your body. So you'll see there's some little lines inside the cell here. That's your cytoskeleton. It's little strands of filament and protein that are holding the cell in a particular shape or helping keep it together. There's also a line somewhere around here. Oh, right here, that is pointing to just plain old cytoplasm. That's the stuff that's inside the cell. It's like a jelly. It's a fluid. Now the rest of the lines are pointing towards parts of the outside of the cell. So if I take a look over here, this top line is pointing to the cell membrane. If we look where it ends, it's ending on a very thin layer that's on the inside of that whole external structure. Then we have cell wall, cell wall. And now if you look, it's like there's two layers of it. The second one would be the cell wall of an adjacent cell. And it's on the picture so that you can see that between one cell and another, like you can see over here, there are pores so that material can be transferred even though there's that giant cell wall in the way. So I'm just going to pause here. So I'm just going to go over this picture of the cell so that you're sure that you identified all of the parts. Up here you have the nucleus, the nucleolus, the nuclear membrane, and the nuclear pores, just like you have on the other side. Over here you have the rough endoplasmic reticulum. Over here you have the smooth endoplasmic reticulum. Somewhere on the rough endoplasmic reticulum you should have seen a ribosome. Now this picture here, like a few people asked me, is the same as the outside of the cell, but it's the zoomed in view. This is simply the cell membrane. An animal cell does not have a cell wall. It only has a cell membrane. Here is a mitochondria, but there are several. Here is your Golgi apparatus. Up here somewhere you can see all the little lines. You could put cytoskeleton. Basically anywhere there isn't something else, like right here, you could put cytoplasm. And once you've done that, there are a few pieces left that maybe you're unsure how to name. For example, you have these little round things. There are just plain old vacuoles, like this one. And then there are vacuoles that obviously have some sort of junk going on the inside. This is an example of a lysosome. This is different. This is new. Animal cells have many of these, whereas a plant cell has few or none, because this organelle is for digestion. Plants do not need to digest because they're making their own food, but animal cells need to take food that they've ingested from some other source and digest it. The other piece that you probably couldn't have identified was the little blue licorice tubes that are crossed one over the other. That's what I think it looks like. This is called a centriole. It's for cell division. Animal cells have them, but plant cells do not. Now, just in terms of knowing which piece has which name, does anybody have any questions that they'd like to ask about that? You would have to know how to name all of the parts of a cell. That is true. Does anyone have any questions they'd like to ask about the names? Yes.
So the little parts of the wall, is that also a vacuole?
This thing right here? Yep, we could say that's a vacuole too. Anybody else have questions that they'd like to ask? Now, am I going to ask you to just name every single part from scratch with no clues? No, that would be ridiculous. If I wanted you to name parts, I would point to them or tell you what their function is. And it's not like any one question is going to ask you to name all of the parts all at once. What we're going to do, now that we know what they're called, is talk about what each of the parts of the cell does. Now, depending on your preference, I know some people write bigger than others, and maybe you have space on your sheet and you want to write the function right underneath the name, or perhaps your writing is like mine, takes up a lot of space, and you want just a new piece of paper. Either way, whatever your preference, get yourselves ready so we can talk about the functions. All right, so there are a few organelles that I'm going to give you maybe a little bit longer of a description of. It's not for every single organelle that I have a lot to say, but for some of them I do. So I'm going to point those ones out first. With each of them on these slides, I've put a really large picture of what that organelle looks like in case you were having trouble distinguishing its appearance. And so I don't think that you need to draw those pictures necessarily, but just so that you can associate what you're writing with which thing on the actual picture it is. So, in no particular order of importance, the first one we'll talk about is the Golgi Apparatus. The Golgi Apparatus, like most of the organelles inside of a cell, is surrounded by a membrane. What it really is, if you look at it closely, is a series of flattened membrane sacs. So if I look at one of these, it looks like it used to be a circle, and then someone stepped on it and it got squished. The Golgi apparatus's job is to synthesize packets, vesicles, or vacuoles, or really any other small little circular pouch that contains things like lysosomes. So the Golgi apparatus is actually just a bunch of membrane all squished together. And what happens with the Golgi apparatus is that vesicles will come in and vesicles will go out. So the idea is that the cell will acquire new things. Those things will be in little sacs surrounded by a membrane. The Golgi apparatus will absorb that bit of membrane and then make a new vesicle using up some of its existing membrane. So basically, it's a spot where I can add membrane and take membrane away. So all that's really happening here, and the way the picture is drawn is so that you can really get the impression the new vesicle is like a piece of the Golgi apparatus pinching itself off. Would anybody like to ask any questions about what the Golgi apparatus does? So if you look and you see something forming, a vesicle or a vacuole forming in a picture, that's your clue that the Golgi apparatus is really close by. Now the other organelle that I'm describing in a bit more detail here is a chloroplast. A chloroplast is something I would find in plants or algae or any other type of organism that is called an autotroph. An autotroph is something that makes its own food. If I look at the word, auto, like the word automatic, means on your own, and troph is referring to your spot in the food chain and how you acquire food. So plants and algae have these. Anything that does not make its own food does not have a chloroplast. They capture solar energy and they convert it to chemical energy via the process of photosynthesis. And I'd just like to make sure everybody knows what photosynthesis involves. Most of the science courses that you would encounter in high school at least mention photosynthesis. Whatever you guys are talking about over there, I can hear you over here, so I can only imagine how it's disturbing the people that are closer to you. In photosynthesis, you take carbon dioxide and water, You use that with the energy from the sun, and you produce glucose and oxygen. That's not a balanced chemical reaction, that's just a skeleton reaction to show you the pieces. But we're taking CO2 and water and converting it into glucose and oxygen. It would be important that you know that basic idea. Now the next couple organelles are lysosomes and mitochondria. So we'll talk about lysosomes first. A lysosome is a membrane-surrounded little sac. So it looks just like a vacuole or a vesicle, but it has different particular contents on the inside. They have hydrolytic enzymes that are needed for digestion, that are needed to recycle material, and that are needed for something called apoptosis. Sometimes cells need to die. They've outlived their lifespan. And so these enzymes can help break apart all of the pieces of a cell so we can reuse them. Now on the bottom, you have a mitochondria. Well, you have a mitochondrion in the picture, but I'm describing mitochondria in general. A mitochondria's job is to capture and then transform energy. It's getting its energy from glucose. So, mitochondria and chloroplast are related to each other. A mitochondria performs cell respiration. It's going to take that glucose that got made during photosynthesis and reverse the process, basically. In cell respiration, glucose and oxygen are going to make CO2 and water and a whole bunch of energy. Now, if I look at a mitochondria, they actually have a double membrane. This point would be discussed later on, like if you're going to take Bio 20, you're going to talk about evolution. It gives us a clue as to how things might have evolved, the fact that it has two membranes. The one on the outside is smooth, so if I look at a mitochondria from the outside, it just looks kind of like an oval. But the internal membrane is curvy. And so whenever we show a mitochondrion sliced open, it's got that wavy line on the inside. The wavy line on the inside is called the cristae, and that's the spot where ATP is produced. That is your energy molecule. The other thing that wavy line does is give more surface area, and that's actually a topic we'll discuss in just a few lessons, so I'm mentioning it now so that when we talk about surface area, I can come back to this point. So just one more thing that I'll mention while we're on this particular slide is that animal cells have many lysosomes. Plant cells have maybe one, or like they have a few of them, but they definitely don't have the number that an animal cell has. And so in your picture, there isn't even an example of one on the plant side. A mitochondrion, like you saw in the picture, is found in both types of cells. And somewhat of a misconception is that if plants do photosynthesis, well, then animals must do cell respiration. But keep in mind, a plant cell has a mitochondria. It's going to use some of the energy that it produces through photosynthesis. And I'm mentioning that because even after several biology classes, some people still have that misconception, or if they don't think about it closely enough, they forget. Before we go on to the next set of organelles, does anyone have any questions that they'd like to ask about lysosomes or mitochondria? Okay. So we'll talk about the endoplasmic reticulum. The endoplasmic reticulum can be smooth or rough. And I already showed you that the difference between smooth and rough is whether or not ribosomes are present. The smooth endoplasmic reticulum synthesizes lipids. Just so we're clear, a lipid is a fat. It's what type of molecule it is. The rough endoplasmic reticulum does a bunch of things. It forms compartments, provides mechanical support, and it produces proteins with the help of those little ribosomes. So anytime I'd like for you to identify which part of the endoplasmic reticulum you're looking at, look for the ribosomes. Ribosomes will always indicate which part is the rough endoplasmic reticulum and which part is the smooth endoplasmic reticulum. Now the next thing on the list is ribosomes. And you'll see that the description for what a ribosome does is really sciencey sounding. It has lots of very fancy words in it. So I'm going to try to simplify it so that you can see what the main idea is. A ribosome is something that's composed of two parts. So there's one, there's the other. Now later on, if you're going to take Bio 30, you will learn at length what a ribosome does and what it is. But on the surface level, what happens is that those two parts fit together. So they interact together to become the site of protein synthesis. So remember, synthesis is just a word for formation. So this is actually where a new protein is formed. The way a protein is formed is by taking your genetic material, your DNA, reading it, and taking from it the instructions for making specific polypeptides, which is what a protein is made out of. And so if I was to look at this ribosome, basically what happens is that your DNA, which is a long, twisty chain, would just kind of run through the middle of it there. It would get read, so it would give instructions to the ribosome, and as that's happening, a protein would get put together. So it's kind of like something that's reading a tape. I know that this is foreign to you because you guys have never seen a cassette tape or videotape, but something that reads like a piece of film would be analogous to what a ribosome is like. So the last thing on this slide is a vacuole. Vacuoles play a role in digestion and waste production. And so a vacuole is actually a pretty general thing. I'm just going to make this move a little bit so we can see it. So vacuoles play a role in digestion and in waste production. They can also be for storage. So if I had to summarize, digestion, waste, and storage are the three things that a vacuole is really known for. Vacuoles can be large or small. They don't have to be one particular size. But notice here that I'm mentioning specifically the huge central vacuole of a plant cell that stores water, because we'll talk about that particular thing a little bit more later on. Does anyone have any questions that they'd like to ask about any of these organelles? So what we'll do then is I'm going to go to my sort of summary page that has all of the organelles on it. And I'm going to show you with just a few words what the function of some of the other organelles is, because I think that I can describe them in a lot less words that I needed for these other organelles that are here. Does anyone still need this screen up? Okay, we'll just wait for a second. This slide sums up all of the organelles. We've already done a bunch of them, Like we already mentioned, mitochondria, chloroplast, vacuole, Golgi apparatus, endoplasmic reticulum, ribosomes, and lysosomes. So the ones that we didn't mention specifically already are the other ones that are on this list. All of the organelles, notice, are compartments with a membrane. And the purpose of having all of these organelles is to localize, or maybe I'll say specialize, cellular metabolism reactions. What we're saying is we're trying to make each thing that a cell needs to have happen in a particular place. So we named on our picture of the cell the nucleus, and its job is to control the cell. It contains the DNA. Now the nuclear envelope and the nuclear pores together allow entry or exchange of material. Now the nucleolus is the central core of the nucleus. It's the part of the nucleus where all of the control is coming from. Now some of the other parts that we needed to mention would be the cytoskeleton. Its job is to provide support. And so if you looked at the pictures, it looked like little strings of fiber or almost like little bones. That's what its job is. The cytoplasm's job is transport. That's because it's a fluid or a jelly and substances can move through it. So it allows things to move from one side of the cell to the other, from the cell membrane in towards an organelle that's going to do something with it. The cell membrane and the cell wall we will discuss in more detail in the next section, and so I'm not going to talk about them very particularly right now. If I wanted to look just at a very superficial view, the cell membrane does a really similar thing as the nuclear membrane, where it allows entry and exit of substance, so exchange to happen between one cell and another cell. The cell wall is made out of something called cellulose. That's the name of the molecule that makes up the cell wall. Cellulose is something that humans have trouble digesting, and so when we say plants have undigestible fiber in them, part of it would be because of this cellulose. The last thing at the bottom, and I'll just make it so we can see it, there we go, are centrioles. Centrioles are for cell division. If you plan on taking further biology courses, you will talk about all of the steps of mitosis and meiosis very specifically, so I won't get into them right now. But a centriole is necessary during cell division to help chromosomes divide themselves up properly. And does anyone have any questions they'd like to ask about the organelles that are found in a cell?